Good morning, viewer. Bienvenue. Here's the most amazing news for our viewers. University of Michigan researchers have made significant strides in understanding and treating a new aggressive subtype of prostate cancer linked to the CDK12 gene. They have developed a mouse model to study the gene's impact, discovered how it induces DNA damages, and created an innovative oral therapy that targets CDK12 and CDK13, showing promise in preliminary tests. This therapy, when combined with existing treatments, could potentially overcome the common problem of resistance in cancer treatment. After scientists at the University of Michigan Rogel Cancer Center first discovered a new subtype of aggressive prostate cancer, they knew they needed to understand how this genetic alteration was driving cancer and how to target it with treatment. In two new papers, each published in the journal Cell Reports Medicine, they do both describing the mechanism of how alterations in the CDK12 gene drive prostate cancer development and reporting on a promising degrader that targets CDK12 and a related gene to destroy tumors. Investigators previously found loss of the CDK12 gene in about 7% of patients with metastatic prostate cancer, suggesting this alteration may be linked to a more aggressive form of the disease. This was discovered from DNA and RNA sequencing from patient tumor samples. Also, CDK12 also plays a role in some ovarian cancers too. To understand how CDK12 loss impacts cells on a molecular level, researchers created a mouse model to try to parallel the genetic alterations they were seeing in human prostate cancer. What was quite surprising was when we created CDK12 loss in a mouse prostate. This caused precursor lesions to form in the mouse prostate. Then, when you added loss of the P53 oncogene, the mice developed bona fide invasive prostate cancer, said senior author Arul M. Chinayan, MD, PhD Director of the Michigan Center for Translation Pathology and S.P. Hicks Professor of Pathology at Michigan Medicine. It will be an addition to the field to have a genetically engineered mouse model that parallels what we see in human prostate cancer. With the mouse model, researchers from the above institution discovered the mechanism of how CDK12 loss induces DNA damage. The loss of this gene activates other known cancers driver genes, causing them to be overexpressed at a high level while also causing DNA to be replicated very rapidly. The coalition of these two processes leads to DNA damage. These back-to-back studies taken together are quite impressive. We created an animal model and then deciphered the mechanism of how CDK12 loss actually drives prostate cancer, Chinayan said. The team also found that a partner gene CDK13 is important in targeting the alteration therapeutically. They developed a potential therapy designed to degrade CDK12 and CDK13. Testing in cell lines and mice show the degraded specifically binds to CDK12 and CDK13 and stops the growth of cancer cells over normal cells. The degrader can be absorbed orally and would not need to be delivered intravenously. This is notably as most protein degraders are too large to be absorbed orally, which has limited their potential in drug development. Behind this exclusive report.